Hello, this is Botany 101 and I'm Angela Nishimoto. Today we're going to do the cell. This is the subject of our lecture, okay? And first of all, we're going to go through the history of the cell, okay? In 1665, Robert Hooke, whose image is here on the screen, was the first to actually look at cells. He was so excited, he used what they call a primitive microscope to look at cork, slices of cork bark, okay? And he compared it to a honeycomb, and he actually called them cells because the cells that he looked at were empty. They were not living cells. Now in the 1670s, Marcello Malpighi, Nehemiah Grew, and Anton van Leeuwenhoek, they looked at various parts of plants and also various parts of animals, and they reported for 50 years on different parts of different cells, okay? And they also noticed and they also observed these single-celled animals or single-celled microbes that look like animals to them, and they called them animalcules. Okay. Now in 1809, Jean-Baptiste Lamarck, who's kind of famous in terms of evolutionary theory, he actually was the very first to state that nothing, no living thing can actually be alive if it's not composed of cells. Okay, so he was the first one to articulate that living things are actually made up of one or more cells, right? Now in 1831, Robert Brown, after whom Brownian motion is named after, okay, he was the first to actually observe a nucleus, and he stated that the nucleus existed in all cells, at least all the cells that they had observed up to that time. Now in 1838, so between the 1600s and the 1800s, there wasn't that much development in terms of cell theory. But in 1838, Matthias Schleiden, who was a botanist, and Theodor Schwann, who was a zoologist, actually formally articulated the modern-day cell theory. That states that all living things are made up of cells, one or more cells, and cells are the bases of the organization of life. Okay. So in 1858, Rudolf Virchow stated, "Omnis cellula e cellula." That means that every cell comes from a pre-existing cell. So he actually tried to state that that spontaneous generation doesn't happen, which was quite controversial at the time, because many people believe that animals could actually be spontaneously generated, okay, from dust, okay, which was something that many people believed. So his statement that all cells come from pre-existing cells was quite a controversy. Okay, so in 1860, there was a prize offered for anyone who could either support the hypothesis that spontaneous generation happens, or to disprove that spontaneous generation happens. Okay, and in 1862, just 2 years later, the great Louis Pasteur, the brilliant Louis Pasteur, did his experiments using broth in swan neck flasks. So he was actually able to show that the broth in the flasks remained clear or uncontaminated as long as they were not exposed to any kind of outside microbes in the air. Okay. Now microscopes. Okay, microscopes are the tools by which we actually look at cells. Okay, because remember, cells are microscopic. Okay, they magnify the images of cells. So light microscopes use light that passes through lenses, and the curves of the lenses help to minimize distortion. Okay, and we have two kinds of light microscopes. We have the dissecting microscope, also known as the stereo microscope, which magnifies about 30 times magnification, and it will actually magnify opaque objects and give you a three-dimensional look at them. All right, like this piece of, it looks sort of like coconut, okay? And then we have the compound light microscope, which uses light. Okay, you can actually get to look at some organelles, like in this section here, Okay, you can actually see some xylem tissue, which is a water conducting tissue, and some parenchyma cells, which is a generalized cell type. So the specimen needs to be thin enough so that light can pass through it. So what we're going to do is we're going to go over to the microscopes here. And here we have a dissecting microscope. Okay, so the first thing you do is you put your specimen in place on the stage, and then you turn the light on. Okay, this one actually has a light control too that can control whether how bright or how dim you want the light depending on your needs. Okay, so this can be lit from the top, or it can be lit from the bottom with this fluorescent light, or you can light it from the top and the bottom at the same time if you like. Okay, so what you'll do is you'll kind of look through the oculars to look at your specimen, and depending on what magnification you have it at, you can actually focus up or down to get it into focus. And I'm looking at the leaf of a succulent plant. Okay. Anyway, so that's how you actually use a stereo microscope or a dissecting microscope. Now, a light microscope, a compound light microscope, you need to actually use a slide, whether it's a prepared slide like this, or if it's one that you make yourself. If you do make the slide yourself, you need to make very, very thin sections. It needs to be able to fit between a glass slide and a cover slip. So they're very, very thin. So remember, you need to actually shine light to the specimen. Okay, so what you do is, first of all, you put it in the clip, okay, and then you can use the coaxial stage controls to get it centered over the condenser. You turn it on, and you also have a light control switch here on the side to make the light more intense or less intense. And my advice to you as you're using the microscope is to use as little light on it as possible. Okay, because if you use too strong of a light, you're probably going to get a headache, okay, if you're viewing something for more than just a couple of minutes. Okay, then you look through eyepieces, which also are adjustable in the compound light microscope. And this one has biocular vision, or has two eyepieces. All right, and you have the specimen on the stage, and then you would use a large knob if you have it at scanning power. You use a large knurled knob to get it into focus. You can use the coaxial stage controls. Then what you do is that if you want it at larger magnification, like at 100-time magnification, then you'd use a fine focus knob to get it into focus. Okay? Because if you use the coarse focus knob when you're at low power or high power, you might send the slide crashing into the lens, and that wouldn't be good. You could actually lose both the slide and also the lens itself could be damaged. Okay, so if you're taking the lab, the use of the microscope is very important. In fact, you will probably be using the microscope quite extensively for at least a few lab periods. So it will behoove you to actually learn how to use the microscope. I mean, don't let just one person in your group do all the focusing all the time. Everybody should have a turn at doing it. Okay, now we're going to go back to the lecture. Okay, so here we are. Okay, continuing with microscopes. There are also electron microscopes that use electrons. All right, a beam of electrons is produced when electricity actually goes through a wire. Okay, now the scanning electron microscope, or the SEM, can magnify about 10,000 times, which is amazing. Okay, and what it does is the electrons will scan the surface details of the cell or the cell structures. Okay, and the beam goes through a vacuum, okay, on both the scanning electron microscope and also the transmission electron microscope, or TEM, which can magnify even more, up to 200,000 times. Okay, so the scanning electron microscope can actually give you a good indication of the surface features of what you're looking at, whereas the transmission electron microscope, the TEM, can actually give you a section of what you're looking at. This looks like a mitochondrion. Okay, So it actually forms an image, passes the electrons through the specimen, and forms an image. Okay, so microscopes are used to look at cells, right? Now there are two main different types of cells in life, okay? We have prokaryotic cells that are cells that do not have a nucleus. Okay, pro means before and karyon means nucleus or kernel, all right? So prokaryotic cells do not have nuclei, right? So here you have actually the genetic material of the cell. Remember we said before that the components of every cell include the cell membrane or the plasma membrane, which you see on this bacterium, okay, cytoplasm, the liquid in the cell with maybe bodies suspended in it, and then we have genetic material, the three parts of every cell, okay? Now eukaryotic cells are cells that do have a true nucleus. Okay, they also have membrane-bound organelles, whereas the prokaryotic cells do not have membrane-bound organelles. Okay, so this cell here, it looks like an animal cell, all right? You can see the cell membrane right here in purple. Of course, it's not really colored this color. Okay, the color is actually added to the illustration so that you can see better. Okay, and here's the cytoplasm with organelles suspended in it. And then here you have the nucleus. It has a genetic material in it. Okay, so that's the difference between a prokaryotic cell and a eukaryotic cell. Prokaryotic cells do not have nuclei, do not have membrane-bound organelles, and the eukaryotic cells do have a nucleus and do have membrane-bound organelles. Okay, now cell structure and communication. Okay, in a plant, you have the cell wall, okay, and the cell wall surrounds the protoplasm, which is the living part of the cell, okay. Now, living plant cells are bounded by the plasma membrane, okay. The plasma membrane, if you recall, is made up of phospholipids, okay, with intermembrane proteins, all right. Now, we're taking a look at the cellulose in the cell walls. Okay, so the cell walls usually are made mostly of cellulose, and this is sort of how they're constructed, sort of like smaller units making up larger units, making up even larger units. All right. Now the cytoplasm, okay, is the cell parts between the plasma membrane and the nucleus. Generally, the nucleus is not considered part of the cytoplasm. It's considered separately, right? And the cytoplasm is made of cytosol, which is a soup-like fluid inside of the cell, and you have in them suspended organelles, okay? Organelles are structures that have specialized functions, okay? Such as the mitochondria, such as the chloroplast, such as the ER, and etc. And don't worry, we're going to go into all of these in just a minute. Okay, so don't worry about it. We're going to go into them one by one, step by step. Okay. Now here we're going to go through the parts of a cell. We're going to do like a preview before we actually go through all the parts of the cell one by one. Okay, so just to get you used to looking at the diagram of a cell, or a diagram of a cell. There's actually a couple of diagrams that we use, right? So here you have the cell wall. Okay, right inside of that is the cell membrane. Okay, so the cell membrane is oftentimes pressed up right up against the cell wall. All right. Here you have the nucleus. Okay, and then you have continuous with the nuclear membrane. You have the ER, the rough ER, and the smooth ER. Okay. Here you have a Golgi apparatus, so don't worry, we're going to go into these one by one in a little bit more detail a little later on. We have the central vacuole, which maintains turgor and pressure inside of the cell for internal support. Okay. Here we have the part that is colored inside of the cell, so these are actually green chloroplasts. And here's a mitochondrion. Okay, here's a microbody called a peroxisome. Okay, and here you have parts of the cytoskeleton. Okay, so we're going to go into the parts one by one as we continue. Okay, so cell size. Most cannot be seen with the unaided eye, all right, because plant cells are only about 10 to 100 micrometers large. Okay, that means that there are 500 plant cells in 1 inch, so that's way too small for you to be able to see without a light microscope. Okay, so why are cells so small? Okay, so smaller cells have a larger surface area to volume ratio. Okay, that means that this large cell here, okay, has a smaller surface area compared to its volume. Okay, so here you have these smaller cells that have a larger surface area to volume ratio. So if you were actually to increase a cell's diameter 10 times, the surface area would increase 100 times, but the volume would increase 1,000 times. Okay, and so that's important because what that allows for is that if the cell is smaller, it allows for faster communications from one part of the cell to another, like from the surface of a cell to the interior parts of the cell, because that's where some substances come in or some communication from another cell happens from the exterior of the cell. Also from the nucleus of the cell, which is the control center of the cell, it actually sends out messages to other parts of the cell faster. Okay? Now the cell wall, okay, it bounds the cell. It provides for external support of the cell, because plants have no endoskeleton or exoskeleton. It actually needs to have external support from the cell wall. Okay, so the cell walls are either rigid support that lends strength, or it has semi-rigid support for flexibility. Okay. Now the cell wall also serves as protection. It kind of helps to protect the delicate structures within the cell. And components of the cell wall, here's another image of a cell, is cellulose. Okay, that's the main component. There are usually about 100 to 15,000 glucose monomers that make up a molecule of cellulose. Okay, another component is hemicellulose, which is like a glue-like substance. Okay, pectins, which is of course used by us humans to make jellies. And then you have glycoproteins, which are proteins that have sugars attached to them. Okay? So now the middle lamella is the area between cells. Okay? It's a layer of pectin between cells. Now the secondary cell wall. Okay, the secondary cell wall is made up of cellulose and lignin. Okay, what it is these cellulose fibrils are embedded in the matrix of lignin. Okay, sort of like rebar is embedded within cement. Okay, so you have these cellulose fibrils embedded in the cement of the lignin. That makes the support very strong and somewhat a little bit flexible. It can take a little bit of a shock sometimes. Okay. So now we have the intercellular connection called plasmodesmata. Okay, so what they do, they actually allow fluids and dissolved substances to pass through these plasmodesmata. They're like tiny strands of cytoplasm, okay, that extend through small openings between cell walls, between adjacent cells. Okay, and they are involved in the movement of sugars, amino acids, and ions. Okay, from one cell to another. Right. Now the cell membrane controls the movement of substances in and out of the cell. Even though the cell wall bounds the cell wall is still pretty freely permeable. Okay, it allows many substances to go in and out, but what actually controls what can get into the actually living part of the cell is the cell membrane. Okay, it controls the movement of substances into the cell and out of the cell. Okay, and it's the outer part of the living part of the cell. All right. So it's semi-permeable. It allows some things through, doesn't allow other things through. Like the cell membrane allows non-charged particles such as carbon dioxide and oxygen gas. Okay, so that's how we actually do our gas exchange in our cells, as well as plants do their gas exchange, okay, through the membrane, right? And it's of course made of the phospholipid bilayer. You can see it here, where the phosphate heads turned outwards and inwards, or inwards and outwards, okay, with intermembrane proteins, or some proteins just at the surface, or some spanning the membrane, okay? And this is what they call the fluid mosaic model. That means that these phospholipids can actually float around, okay? They can actually be removed from the membrane, and when whatever causes it to be removed is taken away, then they can actually reassemble into a membrane, okay? It's a fluid mosaic model, so the proteins can actually, if they're not anchored by the cytoskeleton, they can actually turn over, they can actually go sideways, okay? They actually can float around, so it's like protein icebergs in a sea of lipids. Okay. Now the nucleus, okay, is a control center of the cell, right? It says coded information from DNA to other parts of the cell, okay, they're involved in growth, differentiation, and other activities, okay. It also stores hereditary information, okay. So the nucleus itself is sort of like a sort of roundish or maybe ovoid structure in the cell, okay, and it's surrounded by the nuclear envelope or the nuclear membranes, which is a double membrane, okay, and they have pores in them to allow, say, mRNA to come out or amino acids to go in, okay, or also nucleotides to go into the nucleus, okay. And in the nucleus, of course, you also have nucleoli, sometimes one, sometimes more. Okay, so a singular nucleoli is a nucleolus, right? And nucleoli are made of RNA and proteins, and they're involved in the formation of ribosomes, which we'll get to in a second or so. Okay, and of course, the nucleus actually is very important because it stores genetic material, DNA, in the form of chromatin. Okay, it's usually in the form of chromatin, which means colored threads. Okay, and when chromatin condenses, it forms chromosomes or colored bodies. Okay, and these, of course, are made up of DNA and proteins. Okay, now each species, okay, each species of plant, each species of animal has a particular number of chromosomes. Okay, so radish actually has 18 chromosomes in each of its cell. Okay, a species of golden weed has 4 chromosomes per cell, and adder's tongue fern actually has more than 1,000 chromosomes per cell. And the number of chromosomes per cell doesn't really have anything to do with the complexity of the organism. I think in animals, I think dogs have 78 chromosomes, and we humans, of course, have 46. Okay. Now, the endoplasmic reticulum is continuous with the nuclear membrane, okay, and it functions in communication and channeling of materials from one part of the cell to another, okay, and there are 2 different kinds of ER, or endoplasmic reticulum. We have the rough ER that has ribosomes, okay, and these little things here representing ribosomes are usually grouped together, okay. And what they are involved in is they're involved in the synthesis, secretion, and the storage of proteins. That's what the rough ER does. Okay. Now the smooth ER is called the smooth ER because they do not have ribosomes, so they're smooth. Okay. And they're involved in the secretion of lipids. Now ribosomes, I told you we'd come to these. Okay. They're actually made of RNA and proteins. Ribosomal RNA, okay? And they're actually made in 2 units. You have the large subunit and the small ribosomal subunit. Okay, so you have a binding site for what we call messenger RNA or mRNA, and then we have these sites for transfer RNA or tRNA. Okay, we'll talk about this more later in the semester when we're talking about protein synthesis, because of course the ribosomes actually do protein synthesis. Okay. Now then we also have the dictyosomes. Okay, now the dictyosomes actually form the Golgi apparatus. Okay, so dictyosomes are stacks of flattened discs. All right, and in 1898, Camillo Golgi was the first person to describe and see a Golgi apparatus, so he actually had it named after himself. Okay, and they're involved in the modification of carbohydrates and proteins. Right, they collect and package and also deliver, which is kind of amazing. They do all these different functions. Polysaccharides, all right, flower nectars, okay, and also essential oils. Now we have the plastids, okay. The most important of these plastids, of course, all of them are important, but the most important for the future of the planet are the chloroplasts, okay, which have green chlorophyll and other pigments within them, okay. They have a double membrane around them, all right, and they function in photosynthesis, the most important biochemical reaction on Earth, okay. So generally, you have two parts that are important in the chloroplast. You have these stacks of thylakoid, kind of coin-shaped membranes known as thylakoids. They're stacked in stacks called grana, singular granum, G-R-A-N-U-M, all right? And these are the places where the light reactions of photosynthesis happens, okay, the photochemical part of photosynthesis, okay, where the plant harvests light, okay. His photosynthesis actually converts light energy into the energy of chemicals called sugars. The second part of the chloroplast that's important is a jelly-like liquid inside called the stroma. Okay, the stroma is where the Calvin cycle happens. Okay, and the Calvin cycle involves the fixation of carbon dioxide, and it uses water and carbon dioxide to actually make sugars, okay, chemical energy. Now other plastids include chromoplasts, okay, and these tend to be yellow, orange, or red, okay, and they tend to be the color that they are because of pigments called carotenoids. Okay, so carotenoids can either be more orangey, reddish color, and those are the carotenes, all right, or they can be more yellowish and brownish in color, and those are called the xanthophylls, all right. So what we see here, okay, on the computer is actually looking at the cells probably of red pepper, okay, because the color in tomato, the color in carrots, and the color in red pepper all come from these little bodies called chromoplasts, which have these pigments in them, okay, the carotenoid pigments. Okay, there are also leucoplasts, which are colorless or white. So we have ameloplasts, and this is a section of potato cells which are a kind of leucoplast that store starch, or actually produce starch, synthesize starch. Okay, and then in other cells you have elaioplasts that synthesize oils. Right. Now then we have the mitochondria. Okay, the mitochondria is actually the powerhouse of the cell. Okay, and what it has is, it's another organelle that has a double membrane around it. Okay, the outer membrane is relatively smooth, not that bumpy, but the inner membrane is infolded with cristae, as they call it, cristae, right? And that allows the inner membrane to have more surface area, because in the inner membrane you have these electron transport systems. Okay, when we get into physiology, we'll get into cellular respiration, which is actually what the mitochondria do, at least part of cellular respiration. Okay, and cellular respiration has to do with the breakdown of glucose, okay, to produce adenosine triphosphate, or ATP, which is the currency of energy in the cell. Okay. Now, there are also microbodies in the cell that have specialized enzymes and have a single membrane. One example of that are the peroxisomes. Okay, peroxisomes have enzymes for photorespiration, and photorespiration happens in order for a plant to survive hot, dry conditions. Okay. Then we have glyoxysomes that work in the conversion of fats to carbohydrates, right? And these are common in germinating seeds, okay, where the seed is actually storing fats, converts it to carbohydrates so the seed can germinate and grow. Now we also have the central vacuoles of the plant or cell. Here it is. It can actually take up 90% or more of the cell volume in a mature plant cell, okay? And it's bounded by a membrane called the tonoplast, okay? The vacuoles function in internal support and turgor pressure. Turgor pressure helps keep the cells kind of full of water and kind of rigid, okay? And inside of the vacuole is cell sap, which tends to be slightly acid in pH. And in this cell sap, there are salts and sugars, acids, proteins, and pigments, okay? And the pigments include anthocyanins, okay? So the earlier pigments we talked about, the chlorophylls and the carotenoids, they're actually fat-soluble pigments, right? Whereas the anthocyanins, which tend to be red or purple or blue, they're water-soluble pigments, so they're different in effect. And in fact, the red and red onions and the red, purple, and red cabbage are from anthocyanins, so they tend to be more toward the purple part of the spectrum, okay? Now the cytoskeleton, okay, functions in internal support, okay, as well. So the both the vacuole and the cytoskeleton function in internal support, okay. It's also involved with movement within the cell, okay. Especially highlighted are the microtubules, which function in cell division. And here's a microtubule, okay. And there are the microfilaments also, which helps to allow cyclosis or cytoplasmic streaming inside of the cell, where the liquid inside of the cell moves around, okay. And in the cell there are also intermediate filaments, but the main ones are the microtubules and the microfilaments. Okay, now we finished talking about the parts of a cell, all right? And right now I want to talk about an assignment that is coming due, your Learning Log 1, okay? Your Learning Log 1 is due 1 week from today, all right? And it should be a paper, a formal paper. So because it's a formal paper, you should use, correct spelling, good grammar, understandable syntax, okay? It's a writing assignment, so I'm going to be grading you not only on the content, but also on the form, all right? I actually taught writing at Hawaii Pacific University for a couple of years. I taught argument, research, and writing, so I do know how to grade papers, okay, just to let you know, okay? So your paper is going to have to do with the parts of the cell, which we just went over, right? Okay, so what you need to do is you need to actually take 5 parts of the cell, at least 5 parts of the cell. You can do more than 5 parts if you want to, okay? And you're going to take these 5 parts of the cell and you're going to liken them to 5 parts of some other system, one other system, okay? Say, for example, if you were to liken the cell to a school, okay, you might say like the cell wall is comparable to the fence around the school, that actually bounds the school and protects what's inside, okay? The cell membrane can be like the guard at the school that allows certain things or certain people in, and doesn't allow certain people in. Like there might be some people that might be completely legitimate to go to the school, and they always say go report to the office too, right? So, the school guard might be the cell membrane, okay? Then you could say the mitochondria, which are the energy breaking down part of the cell, can be likened to the cafeteria, where the students get energy from eating their food. Okay, and you can say that maybe the chloroplast can be likened to maybe the workers in the cafeteria that actually produce the food and make the food. Like in photosynthesis, the chloroplast actually harvests the light and convert it into energy in the form of sugars. Okay, so the cafeteria workers would actually take the raw food ingredients and actually turn it into edible food that the students can eat and get energy from. Okay, and they're eating it, and that could be respiration. Okay, so that's 4 parts of the cell so far. Then you could say something like the principal could be like the nucleus of the cell, the control center of the cell, or you could see even the main office could be the control center of the cell, that actually all orders to the people that work at the school originate from the office or from the principal, depending on who you want to have as representing the nucleus, all right? And the principal or the office is made of components, sort of like the nucleus is made up of the chromatin plus the nucleolus and the nuclear membrane around it, okay? So you can see that the components of the nucleus are like the components of a person or the components of an office. You should actually take this assignment and have fun with it. Even though I'm going to be grading you on grammar and on spelling and like that, just do a good job, and it should be fine, okay? So then what we're going to do is we're going to actually have you turn it in 1 week from today. And the paper should be about 1.5 pages, double spaced. Okay, it can be more, okay? It can be a little less, but you should have enough words in it and enough explanation in it to actually explain the cell pretty thoroughly and its parts, okay? So you're going to compare the cell to one of the system, the 5 parts or more of the cell to 5 parts or more of the other system. Okay, and it's due 1 week from this telecast, all right? Okay, now let's look at cellular reproduction in the next slide, okay? So in cellular reproduction, you have something called the cell cycle, okay? And it's made up of interphase, this part here, and mitosis. So the cell spends most of its time in the interphase, either 90% or more of its time is spent not dividing, okay? Now the interphase is actually divided into 3 parts. You have the G1 phase or the gap 1 phase, okay, during which the cell will increase in size. This is like right after it is divided. So during the interphase, the gap 1 phase, the cell increases in size. During the S phase or the synthesis phase, that's when DNA is synthesized. So what actually started off as a finger-shaped chromosome actually duplicates itself, and then you have an X-shaped chromosome joined at the centromere. Okay, so the chromosome will have duplicated all of its genetic material and made a sister chromosome, so you actually have a chromosome that's sort of like this, where you actually had one like this before, okay? Now during the G2 phase or the gap 2 phase, the organelles replicate and the microtubules are readied to actually form the spindle, and the chromosomes start to condense, okay? Now mitosis is actually nuclear division, all right? So we have prophase, and so in prophase you have the chromatin condensing and coiling to form chromosomes. So this on the right is actually a photograph of a cell, a stained and cleared cell. Okay, you can see the nucleolus here. Okay, and so the chromosomes are condensing and getting thicker. Okay, and this drawing is a good drawing of it, showing the chromosomes. And another thing that happens during prophase is that the nuclear envelope starts to dissolve and the nucleolus starts to disintegrate, okay? Now during metaphase, okay, you must remember that during mitosis, this is a dynamic process. Okay, so we're not actually only static, only in one phase, but it's actually a process that happens. Okay, so during metaphase, the chromosomes will align themselves at the equator of the cell. Okay, so they're all sort of looking like an eyelash or looking like a spider. Okay, so they're at metaphase, which means middle. Okay, the chromosomes are as thick as they can be, and they will be located at the equator of the cell. Then anaphase, okay, remember that the chromosomes are made up of two parts, the two sister chromatids. We're doing anaphase, the sister chromatids separate from each other. Okay, so all the sister chromatids on this side are exactly the same as the sister chromatids on this side for each one. Okay, so what they do is they'll separate with a spindle attached to the centromeres. Okay, so they're pulled apart from each other to opposite poles of the cell during anaphase. Okay, now during telophase, okay, the genetic material diffuses back into chromatin. Okay, the nuclear membrane begins to reform. Okay, and we also have the fact that the nucleolus becomes parent again. Okay, so that's actually part of mitosis. Okay, then after mitosis, you have something called cytokinesis, which means cell division. Cyto means cell, and kinesis means to cut. Okay, and during cytokinesis, you have the formation of the cell plate. Okay, the formation of the cell plate, which will divide one cell into two. Okay. Now we have a slide that has to do with plant cells versus animal cells. So of course, animals have an internal or external skeleton. Okay, and of course, animals move. So that's one reason why they have no cell walls, because if they did have cell walls, it would be too rigid to be able to move. Okay. Also, animal cells divide by pinching in two, right? You have a whole bunch of proteins that actually form like a little lasso around the center between the two new cells. It kind of pinches the two cells into two. Animal cells also have centrioles, which aid in cell division. Okay, they're actually attached to the spindle fibers, which are made up of microtubules. Okay, and there are no plastids, okay, within animal cells, and also no central vacuole. Okay, animal cells do not have central vacuoles. Okay, let's review what we covered today. All right. First, we went over the cell theory, and you recall that it was Robert Hooke in the 1600s who first saw a cell. All right, and then from the 1600s to the 1800s, there wasn't that much development in terms of the cell, even though they did see those, microscopic, so-called animalcules, right? So it wasn't until the development in optics in the 1800s that you had more developments in looking at cells and looking at the substructures of cells, the parts of the cells. So then you had omnicellular, acellular, that all cells come from other cells, right? And then you had the cell theory that was articulated by Schleiden and Schwann, right? That all cells come from pre-existing cells, and that cells are the basic unit of life, okay? The next thing we talked about is plant cell structure. Okay, and some of the things that make plants different from animal cells, of course, plant cells different from animal cells, are that plant cells have a cell wall, whereas animal cells do not, because animals move, right? And plant cells have chloroplasts, because plant cells do photosynthesis. And of course, animals, we eat our food. We don't actually need to make our own food, right? And then the plant cells have central vacuoles, and animal cells do not. The central vacuole is for maintaining turgor and internal pressure and internal structure to the cell, okay? And then we talked about the cell structures and organelles. Okay, so we talked about the cell wall, right, which bounds the cell. We talked about the plasmodesmata, which is an intercellular connection between one cell and an adjacent cell, right? We talked about the cell membrane that controls what can get into the cell and what leaves the cell. We talked about the control center of the cell, the nucleus. Okay, we talked about the ER, the rough ER producing proteins and the smooth ER producing lipids. We talked about ribosomes, where protein synthesis happens. We talked about the Golgi apparatus. Yeah, I have to look at a list because we covered so many things. We talked about the Golgi apparatus, right, which actually processes and packages cell materials. We talked about the plastids, the chloroplasts that do photosynthesis, the chromoplasts that also have a role in the plant in terms of pigmentation. We talked about the leucoplasts, right, which store starch or oils. Okay. We talked about the mitochondria, which are the powerhouses of the cell that do cellular respiration. We talked about peroxisomes and glyoxysomes. We talked about the central vacuole, and we talked about the cytoskeleton. So that's actually a lot of things for you folks to have to remember, okay? And we also talked about the cell cycle and mitosis, okay? The interphase, the mitosis, which is prophase, metaphase, anaphase, and telophase, right? And then cytokinesis, of course, the cell cycle being made of the interphase of mitosis, and the interphase being comprised of the gap1 phase, in which the cell increases in size, the S phase, the DNA synthesis phase, where DNA is replicated, and then the gap2 phase, where organelles replicate, and the microtubules get ready for mitosis, okay? And so what we did is we talked about all those things. There's a lot of for you to remember. Okay, so please, let me reiterate, please do not hesitate to contact me. Okay, call me during my office hour, or you can just call my number and leave a message and I'll call you back. Okay, and let's see, you can email me if you have any questions, if that's an easier way for you to get to me, because during the weekdays I am definitely available every day through email. Okay, or you could come and visit me in my office hour. Okay, I'm always glad to have students come visit me. Okay, anyway, so that's all we have for you today, which is a lot. I know it's a lot, and I sympathize with you. Okay, but you can do it. I know you can do it. You can actually know these things, memorize them, and also recognize them when you see them on an exam. Okay, so that's all we have for you today. Okay, so we'll see you next time. Thank you for your attention, and aloha.